Hello everyone. Today we are going to learn about the techniques of micropropagation. In our previous lesson, we learned about what is micropropagation and what are the advantages of vegetative micropropagation. Now here we learn about how the micropropagation exactly works. So micropropagation involves three stages basically, but some of the authors have also given the stage 0 and stage 4. As we all know that micropropagation is a complicated process, it involves the following stages. Now we'll learn one by one what are these stages. The stage 0 is the initial step in micropropagation and it involves the selection and growth of stock plants, which is for about 3 months, and it is under controlled condition. In the stage 2, what happens is the initiation and the establishment of culture takes place in a suitable medium. Selection of appropriate explant is also important in this step. So the most commonly used explants are organs, shoot tips, and axillary buds. Now the chosen explant, whichever we choose it, we have to surface sterilize it and we have to wash it before use. So the stage 1 involves the initiation and the establishment of culture. The second stage is the stage where the activity occurs in a defined culture medium. Now it involves the multiplication of shoots or rapid embryo formations from the explant. This is the stage 2 process. The stage 2 process, the multiplication of shoots takes place basically. In the stage 3 process, what happens is the transfer of shoots takes place to a medium for rapid development into shoots. The shoots are directly planted in soil to develop roots. Now, in vitro rooting of shoots is preferred while simultaneous handling of large number of species also take place. So this is stage 3. That what you have to remember here is that transfer of shoots takes place to a medium for rapid development into shoots. In stage 4, it involves the establishment of plantlets in soil, and this is done by transferring the plantlets of stage 3 from lab to environment of greenhouse. Now, some of the plant species, stage 3 is skipped and unrooted stage 2 shoots are planted in pots, and then it is or in a suitable compost mixture. So this is the stage 4 where the transfer of plantlets to sterilized soil for hardening under greenhouse environment takes place. Now the different stages are useful for comparison between the two or more plant systems. So it is important for us to study the different plant system, and it helps in our better understanding. So all the species need to be propagated. It is not necessary that all the species should be propagated in vitro through all the 5 stages, but basically the micropropagation method, this is the basic process which is carried out where the selection of mother plant is done, then the establishment of culture takes place, then the multiplication of shoots takes place. These are the basic steps which are involved. Now the micropropagation involves in vitro clonal propagation by two approaches. Now these are the approaches where the multiplication by axillary buds or apical shoots takes place, or by the multiplication of adventitious shoots. Now besides these two approaches, the plant regeneration processes, organogenesis and stomatic embryogenesis may also be considered as micropropagation. So we'll learn about them in bit detail further. But what exactly is organogenesis is? It is the formation of individual organs such as the shoots, roots directly from the explant which lack the meristeme, or from the callus in cell culture which is induced from the explant. And the stomatic embryogenesis, it is the process where the regeneration of embryos from stomatic cells or tissues and organs takes place. So we learn that further in more detailed manner. So this is what is organogenesis and stomatic embryogenesis. I have already told you that organogenesis, it is the formation of individual organs where the roots or shoots are directly taken from an explant where meristeme is lacking. And the stomatic embryogenesis is where the regeneration of embryos from stomatic cells, tissues, or organs takes place. Now remember that in organogenesis, the most important thing is the callus is also formed over here. So it might be considered that shoots, roots, which are taken from the explant or from the callus, and then the cell culture is induced from the explant. So there are two ways of organogenesis. One is direct organogenesis and other is indirect organogenesis, where one involves the callus formation and the other does not involve the callus formation. Now, coming to the multiplication by axillary buds and apical shoots. Now, actively dividing meristems are present at the axillary and apical shoots. The axillary buds are also located in the axils of leaves, and they are capable of developing into shoots. Now, in vivo state, what happens is that limited number of axillary meristems are there, which can form shoots. Now, by means of induced in vitro multiplication and propagation, it is also possible to develop plants from meristem and shoot tip cultures and from bud culture. Now, apical meristem, it is nothing but a dome of tissue located at the extreme tip of the shoot. The apical meristem, along with the young leaf primordia, constitutes the shoot apex. So, from the development of the disease-free plants, meristem tips should also be considered. Now, meristem or shoot tip is isolated from a stem by a V-shaped cut. Now, the size frequently is 0.2 to 0.5 millimeter of the tip, which is critical for culture. And in the larger the explant shoot tip, the better are the chances for the culture to survive. So the good results of micropropagation of explant should be taken from the actively growing shoot tips, and the ideal timing is at the end of the plant's that is the dormancy period. Now the media which is used, so the most commonly used media is the MS medium and white medium. Now these are the two basic media which are used in this culture. Now here also what happens, there are different stages, that stage 1, stage 2 and stage 3. So coming to the stage 1, the culture of meristem is first established. Then the growth regulator is added, which is cytokinin and auxin. And the cytokinin and auxin, why it is added? For the development and growth takes place. So the stage 1 where the establishment of culture takes place and the cytokinin and auxin is also added. Now in the stage 2, what happens is the shoot development takes place along with the axillary shoot proliferation occurs. Now the high levels of cytokinins are required for this purpose. In the stage 3, what happens is it is associated with rooting of shoots and the further development of the plantlet takes place. The root formation is also facilitated by the low cytokinin and high auxin ratio. Now what it means that the low level amount of cytokinin is added and more amount of auxin is added, so that the shoot development takes place at a faster rate. Now this is opposite to the shoot formation, since high level of cytokinin is required in stage 2, where the stage 2 medium and stage 3 medium should be different in composition. So in order to have different composition in stage 2 and stage 3, first in stage 2, what it is done that the high levels of cytokinins are added. But in stage 3, what it is done that the low level of cytokinin and high level of auxin ratio is done. So the optimum temperature which is required for the culture is in the range of 20 to 28 degrees Celsius, and lower light intensity is more important because we have to get the better micropropagation. So in order to get the good micropropagation, these conditions are very important. So from the figure you can get that first what it is done that the explant is taken, shoot tip or meristem, then it is established into the culture, Then further the shoot development takes place where we add the growth regulator. In the stage 3, the rooting of shoots takes place, and further what happens is the rooted plant, it is then taken out, and then it is taken to the green environment where the in a potted plant, so that the growth takes place nicely. So this is the basic of the shoot tip culture, that how the shoot tip culture takes place. Now coming to the bird cultures. Now the plant birds, they have the active meristems which are dependent on the physiological state of the plant. Now there are two types of bird cultures that are used. The one is the single node culture, and the axillary bird culture is the second one. Now in the single node culture, it is very simple. What happens is it is the natural method for the vegetative propagation of plants, both the conditions, in vitro and in vivo conditions. Now here the bird found in the axile of leaf is comparable to the stem tip. Now it is for its ability in micropropagation. Now the plant is taken, a bird along with a piece of stem is isolated, and then it is cultured to develop into a plantlet. So first, from the figure you can get that the plant from plant X plant is taken, and then the node is isolated and cultured. Then further the shoot development takes place and the rooting of shoots takes place. So this is a simple technique. Now in micropropagation, the basic is very simple, that it involves the 4 stages. The basic is simple, but the difference is that in some, the amount of growth regulators that are added, that are different, and sometimes the, like in bird culture, a bird is taken as an explant, whereas a shoot culture, a shoot stem is taken as an explant. So this is the basic difference that you have to remember. Now closed birds are used to reduce the chances of infections. So this is the single node culture, and in single node culture no cytokinin is added. Now coming to the axillary bird culture. Shoot tip along with the axillary bird is isolated. So a shoot tip with the bird, axillary bird is the explant, and then the culture are carried out with high concentration of cytokinin. And in from the figure you can get that the explant is taken, and then the shoot tip is isolated. Further, what happens is the shoot development takes place, then the cytokinin is added. So the high level of cytokinin is added in this, and axillary branching takes place further, which allows the rooting of axillary formed shoots, and then it is easy to develop a plant. So the cultures are carried out with high cytokinin concentration, and the apical dominance stops basically, and the axillary birds develops. Now a good axillary bird culture, the cytokinin auxin ratio which is maintained is 10:1, and it is the, you have to remember this ratio, that 10:1 is the ratio which is used for growth regulator of cytokinin and auxin for axillary bird culture. And the variable, and it depends on the nature of the plant species and the development of the explant use. The explants which are taken, it requires less cytokinin compared to the adult explants. Now the presence of the apical meristem may interfere with the axillary shoot development. So in such a case, it has to be removed. So this is the basic of the axillary bird culture, that how the axillary bird culture is done. Now there is a multiplication of adventitious shoots that also takes place. The stem and leaf structures that are naturally formed on plant tissues, which are located in sites other than the normal leaf axil regions, are regarded as the adventitious shoots. And there are different adventitious shoots that include the stem, bulbs, tubers, and rhizomes. So the adventitious shoots are useful both for the in vivo and in vitro micropropagation. And the meristematic regions of the adventitious shoots can induced in a suitable medium to regenerate the plants. Now, in order to carry out the micropropagation, there are some factors that affect the micropropagation. So, in order to have the successful micropropagation, optimization of several different factors is required, and like we learn them one by one. The first one is the genotype of the plant. This means that the selection of the right genotype of plant species by screening is necessary for the improved and better micropropagation. Plants with vigorous germination and branching capacity can also be used for the suitable micropropagation. The second one is the physiological status of the explant. The explant, that is the plant materials from more recently produced parts of the plants, help and they are more effective than those from the older regions. Now, the good knowledge of the donor plants of natural propagation process with special reference to the natural growth and the seasonal influences will be useful for the selection of the explants. Now, third one is the culture media. The standard plant tissue culture media suitable for the micropropagation for the stage 1 and stage 2 is very important. Now, stage 3 has got certain modification where sometimes like the growth regulators ratio, high concentration or low concentration is required. Now, the minerals are also added. Mineral composition is also required, and it is largely dependent on the type of culture. Now, it depends like which culture we are doing, whether we are doing it a meristem culture or a bird culture, depending on that the culture media required and the um, growth regulators and their composition depends on that. So coming to the culture media there it has got the cultural environment, sorry. So the light and temperature, these are the two important factors. Now light, photosynthetic pigment in cultured tissues do absorb lights and influence micropropagation. The quality of light is also known to influence in vitro growth of shoots, and for example such as the blue light induced bird formation in tobacco shoots. Now the variation in diurnal illumination also influence the micropropagation. Now the illumination is generally 16 hours day and 8 hours night, which is satisfactory for the development of shoot. Now the temperature which is required, the optimal temperature is also important factor. So it is around 25 degrees Celsius. Now there are some exceptions such as the begonia hybrid where requires the temperature, it is 18 degrees Celsius. So it is requires the low temperature compared to the other plants. Now the composition of the gas phase, like the constitution of the gas phase in the culture vessels also influences the micropropagation. The unorganized growth of cells is generally promoted by the ethylene or the oxygen or carbon dioxide, ethanol and acetaldehyde. So that also depends. So three factors that depends on the culture environment are the light, temperature and the composition of the gas phase. Now there are also some factors that are required in vitro rooting. Now for an efficient in vitro rooting during micropropagation, the low concentration of salts, that is the reduction to half to 1/4 from the original, is advantageous. The induction of roots is also promoted by the presence of the suitable auxine. So this is about the basic factors that are required for that affect the micropropagation. So in order to get the good results, we have to consider these factors. Now coming to organogenesis, we know that it is a process of morphogenesis where the involvement of the formation of plant organs, that is shoots, roots, flowers, buds from explant or cultured plant tissue is done. So there are two types of organogenesis. One is direct organogenesis and the other is the indirect organogenesis. Now coming to the direct organogenesis, tissues from leaves, stems, roots can be taken and directly to produce the plant organs. Now in direct organogenesis, the tissue undergoes morphogenesis without going through a callus or suspension formation. Now from the figure you can get more clearly that from a plant, a explant is taken leaf, and from this explant what happens is the callus formation does not take place, or suspension culture medium stage is not there. So the term direct adventitious organ formation is used for the direct organogenesis. Now, further, the induction of adventitious shoot formation directly on roots, leaves, and other various organs of intact plants is used for plant propagation, and it is particularly used for the herbaceous species. So it is useful for the herbaceous species, and the appropriate organogenesis culture system, the exogenous addition of growth regulators, that is the auxin cytokinin, is required, and the concentration of growth promoting substance depends on the age and the nature of the environment besides the growth conditions. So plant is taken, then the explant is taken. From the explant, the auxin cytokinin growth regulators is added. It undergoes the somatic embryo formation without undergoing the callus formation, and then the root plantlet with shoots and roots are formed. And further, it is taken into the environment, green environment, for the formation of the plant. So this is the direct organogenesis. Now coming to the indirect organogenesis, when the organogenesis occurs through callus or cell culture suspension, you can see from the figure, it is regarded as the indirect organogenesis. Now the callus growth can be established through various explants. Different explants can be taken, like leaves, roots, cotyledons, stems, flower petals, for the subsequent growth of organogenesis. Now, the explant for good organogenesis should have the mitotically active immature tissues. Very important, remember that the mitotically active immature tissues are taken in order to have the good organogenesis. Now, the bigger is the explant, the more better is the result, and the chances of obtaining good viable callus and cell suspension culture. Now, it is more likely to select the meristematic tissues, that is shoot, tip or leaf and petiole, for efficient indirect organogenesis. Now, why it is important? Because it gives more growth rate, and the survival rate is much better than the other explants taken. Now, for indirect organogenesis, culture also is an important factor. So, a liquid medium, it can be grown into a liquid medium or solid medium. So, there are two type of medium, like liquid medium and solid medium. So both can be taken. Now the MS media, which is mostly used in tissue culture, it can be taken, or the B5 media or white media. So it can be used in organogenesis. Now the concentration of growth regulators in medium is also critical for organogenesis. Now the varying concentrations of auxin and cytokinin, it depends on the nature of the plant. In vitro organogenesis can also be manipulated. Now it can be manipulated by the low auxin and low cytokinin concentration, which helps in the callus formation. Then the low auxin and high cytokinin ratio, where it will help to promote the shoot organogenesis from callus, and also the high auxin and low cytokinin concentration, which helps in root formation. So the nature of the plant, depending upon that, in organogenesis, what is done is the growth regulators, like in different stages. Now for the formation of the callus formation, we'll take low auxin and low cytokinin ratio.